Next up, I'm excited to welcome up Jon Rask. He's a friend of the state of North Dakota. He's a North Dakota native, UND alumni, and he is a NASA Ames research with NASA Ames Research Center, and is a research scientist who has done a lot when it comes to space biology missions. So, with that, Jon, as a friend of the Space Agriculture Conference, I will turn it over to you.
Thanks very much, everybody. Appreciate the opportunity. Today, I'm here to talk to you about lunar regolith and features of it that are relevant to space agriculture. And what we've heard today is that when you take life from Earth into space, it changes. And the changes are conditional upon the environmental forcings, but also the material that the life may be exposed to. And if we take life to the moon, it's going to be exposed to dust, lunar dust. And this presentation is intended for us to imagine we have the capability to get to the moon. If we're going to grow plants hydroponically, or if we're going to grow plants in lunar soils, what to expect and what to anticipate in terms of what the dust might be doing to the life forms. So in the past, you've heard me talk about my roots in North Dakota and interests in space exploration. I'm shifting a little bit today to talk about how space ag is at the core center of all these astrobiology, space biology, human research, and planetary protection disciplines. This is an awesome interdisciplinary exercise, and I applaud the state of North Dakota and all the partners that are engaging in this effort. No matter where we go in the solar system, our nearest planetary neighbors are full of dust. And that dust is going to have an impact, whether you're on the moon or on Mars. And what I'd like to do is focus on the moon and give you a little bit of a background on the processes that form the moon. And if one of you could cue up the, thank you. So the moon formed from an Earth-Mars sized two objects colliding about 4.5 billion years ago or so. The moon essentially started to cool and crystallize. But then a series of impacts occurred early in its history, most notably the South Pole Aitken Basin, which is the site of the Artemis missions we expect is going to be of great interest. And it's in that location where there's cold traps of a variety of different ices and other types of volatiles. And since then, the moon has experienced huge amounts of impacts that formed the very dark areas on the moon, which we know as the mare, or the basalt-rich sort of floodplains of basalt that are smeared across the surface. And when those mare formed, they all sort of condensed and crystallized at different rates because of the nature of the heating and the type of chemistry that formed the rocks. But once those rocks began to form, they created the surface that we largely see today that has been modified by continuous and constant impact processes. And those impact processes have ground up the surface of the moon into a dramatically various and heterogeneous surface that is, in most cases, up to 20% respirable sized particles. That means everything from 5 microns and smaller. So the bulk of the lunar regolith is extremely fine-grained material. And so up to about a billion or so years ago, we essentially see the formation of the surface that we see today. And we can pause there. I will go to the next slide. So just to recap, the processes that are operating on the surface of the moon are producing and creating what we call the lunar environment. And that includes UV radiation, hard UV from the sun. And that UV is known to be able to alter the chemical reactivity of dusts here on Earth. We demonstrated a paper that we can make lunar simulant more chemically reactive if we expose it to hard UV. In addition to the solar impacts, we see galactic cosmic radiation bombarding the surface, and along with that, micrometeorite bombardments, which is constantly churning up material on the surface, freshly fracturing the rocks to further grind down and pulverize the surface into a fine powder. And to sort of put this into a cartoon, during those impact processes, the micrometeorites that strike the surface are coming in at such velocities that they basically vaporize certain very small regions of the impact site. And then that material, after it's vaporized, immediately condenses and then reforms as solid material on the surface in the form of agglutinates, glasses, and other jagged particles. And common to most lunar particles that are on the surface of the moon, they are studded with elemental iron that has formed from this vaporization and condensation impact process. And that elemental iron is what makes lunar dust magnetic. So if you took a magnet to the surface of the moon, you could move lunar dust around because of the presence of these elemental iron globules. And looking closely at the content of the material on the moon, we don't see a lot of nitrogen, potassium, or phosphorus, those things, or carbon, of course, for those things we need in plants. So if we want to grow plants, we're going to have to probably bring some of that with us. So straight away, I can imagine that this is going to be a challenging material to grow plants in, and I'll touch upon that a little bit later. So how do we know this? Well, there's been 10 sample return missions that I'm aware of. The 6 Apollo missions, the 3 Russian missions, and the most recent China mission that has brought samples back to Earth, and scientists have studied the material and concluded that there's a various chemical makeup of the specimens, and many of the studies performed on Apollo specimens demonstrate that the material which has more elemental iron in it is more chemically reactive. And in fact, we're able to see that there are parts of the moon's polar regions that have actually experienced some small, faint amounts of rusting because of the presence of small quantities of water reacting with that elemental iron to form hematite. So what does chemical reactivity mean in a biological sense? Essentially, the concept is quite simple. Mineral grains will produce these radicals, or reactive oxygen species, which include hydroxyl radicals, superoxides, and peroxides, all of which are harmful to biology. And this is normal. This is expected even in dust chemistry here on Earth. But biological systems have a way to manage that. But if there's too much of it, it can be very harmful. So biological exposure to mineral grains and the chemical reactivity itself elicit an inflammatory response in cells that leads to more production of these reactive oxygen species, which are harmful. So in a nutshell, you can increase reactivity of a dust particle by exposing it to these environmental conditions that we see on the moon, but you could also reduce the chemical reactivity by baking it or exposing it to water and oxygen. So what does this mean for biology and space ag? So these radicals, if they're on the mineral grains, they may be contributing to cell death, problems with cell signal transduction and disruption and their ability to communicate. It's going to alter gene expression, and it may cause oxidative damage to nucleic acid, proteins, and lipids. Now, this is not a doom and gloom scenario. I see this as an opportunity because recently scientists have grown plants in actual lunar specimens and done a number of inflammatory pathway studies and demonstrated that, yes, we can grow them, but the plants are stressed. And so we have to continue this line of research to better understand what we need to do to optimize growth of plants in lunar soils if we're going to use lunar soils. And in addition to that, this dust is also harmful to engineering systems, as you probably heard earlier today. So engineering tests, just like biology tests, must be conducted on appropriately treated specimens or specimens that are freshly collected from the moon. So I challenge and invite everybody that's part of this conference to explore development of designer space egg species that are specifically tuned to growing on the moon to partner with government and commercial launch providers that get us to the lunar surface. And while we're doing all that, let's request some lunar samples from NASA to use in plant studies, conduct the studies that expose the plants to the actual lunar specimens. And while we're doing all that, invent lunar payloads that grow plants on the lunar surface and freshly collected materials. So that involves operational scenario development, as well as ground studies and controls that are done in collaboration with actual flight experiments. So with that, I once again say thank you very much for this opportunity. And I look forward to collaborating with anyone in the future.